and it's my great pleasure to introduce our speaker today, Dr. Julija Krupic. Julija completed her undergraduate degree in physics at Vilnius University in Lithuania, followed by masters in bioimaging sciences at Imperial College London, where she started to apply physics to biological problems working on cerebellar Purkinje cells. Then she completed her PhD in neuroscience at UCL in Professor John O'Keefe's group, working on the medial entorhinal grid cells. After that, she did a postdoc at UCL and at the Salk Institute, and then moved to University of Cambridge to establish her own lab. Her major interests lie in understanding the entorhinal hippocampal circuitry and its role in navigation and spatial memory in health and disease. And recently, her lab has developed a home cage monitoring system which incorporates the TEATIME and novel object recognition tests in the home cage environment in a completely unsupervised way. And today she promised to give us the overview of this novel system called Smart-Kage. So we welcome you again, Julija, and the screen is yours, please.
Thank you very much. Thank you very much, Vootele. Thank you very much, everybody, for your interest coming. And I just said to Vootele, it's my first webinar, so bear with me. Hopefully it will go smoothly. So yeah, I'm very excited to present a new system, home cage monitoring system for cognitive and phenotyping, and also phenotyping of other behavior in a home cage environment for mice. I will start with declaration of interest. So since we developed this technology, we also founded a startup at the University of Cambridge, which develops this kind of technology for neuroscience community. Okay, but then I will first start with something that Vootele kindly already introduced, which is what is my day job, what I'm really interested in. And this is understanding the medial entorhinal cortex and hippocampal circuitry, and especially in the context of both of them. This is how I started, and this is a continuous research area in my laboratory shown in here at Cambridge. And in short, we use very simple environments, actually, most of the time, or at least in the beginning. We record from hippocampus, shown here in yellow, in mice and rats, and medial entorhinal cortex, shown here. We use both electrophysiology and imaging to record from cells, which response curves look something like that. So here, what you can see is the firing activity of one cell called place cell, and warm colors represent a position where the cell is firing, and blue represents position where it's not firing. And grid cells are other cell types in now entorhinal cortex, which fire in multiple fields like that, arranged in hexagonal symmetry. So this is my usually it's an old way of doing it before deep lab cut came about and some other cool technologies. We would put LEDs above animals' head, and we would track animals' position and facing direction. And so that in this way we could also know its velocity at any given point, and then we can characterize these curves in this open space arena. And my focus was actually to investigate how geometry changed these patterns. But then it's also, probably most of you know, that these areas, hippocampus and entorhinal cortex, are extremely important for Alzheimer's disease. So these are one of the first areas which become impaired during Alzheimer's disease. So therefore, it's important to understand what's happening then if we are interested, for example, in the onset of Alzheimer's disease. And this became my lab's interest in the last few years after I started at Cambridge. And so in this way, when we started more translational work, once we started working on translation more, looking at hippocampus and entorhinal cortex in more translational context, it became clear that using this simple open arena test is not sufficient for the questions that we want to ask, because we really were interested in how memory is affected in the early stages of the disease. And this is this beautiful paper from, well, almost 10 years ago, sort of explaining and putting it up front what kind of tests are important to do if we really want to fully characterize at least even like cognitive aspects of various animal disease models. So if we're interested in memory, and that includes T-maze or Y-maze, sometimes used novel object, bodies water maze, radial arm maze, and spontaneous alternation. There are other tests as well, but these are probably the most frequent ones. But what you will notice as well is that how long it takes. And we noticed that too. So one can test any given researcher, no matter how courageous and brave you are and how hardworking you are. On average, you test 10 animals, 10 mice per day in our experience. And the groups that would be required to test, unlike the basic signs that I'm usually was familiar with, it escalates pretty fast, you will see. So we have a controls versus mutant in our case. We have males and females, for example, in UK, as of last year, I think, as recommended by MRC, we can't look just at a single gender, so we really should study both. There are differences and important ones. At least 2 ages tested when it comes to at least Alzheimer's disease. So you can see that to satisfy all these conditions for a single sort of cohort, you would have to test 80 mice a day for 8 weeks, right? And this does not include testing of potential treatments, which I'm also interested in, and it does include replication, and it also assumes that there is only one control versus one mutant. But actually, when it comes to Alzheimer's disease, there is no mouse which has an Alzheimer's disease. So we are using various mutants to simulate or to model various aspects of this disease. So it escalated pretty fast for us. So we needed some kind of a solution to not do all this range of testing, which are important tests to understand where animal is in its cognition journey, so to speak, in the disease progression. So we started looking at various more automated ways, and this is quite old now, one of the first papers, I believe, which actually looked at automatically quantifying behavior in animals' home cage environment. And you can see the brief overview of how it worked. So it was pre these machine learning techniques. It was still image subtraction technique to follow each individual animal, then to analyze these patterns without any kind of deep machine learning protocols, and come up with the classifiers which would assign or annotate various positions, various motion features into categories, which is hang, rear, walk, move, groom, eat, drink, rest. So all of them you will notice. So I will just play you very quickly the video, how it would look like. So you can see it's a very, very simple home cage environment, which would allow you to characterize all these behaviors, but you will notice also that all of these behaviors are passive. So it really would not help me to address this need for testing animals' memory in the case of Alzheimer's disease. So there is another solutions, actually commercially available. So I will say one of the most prominent, I guess, one is called IntelliCage, and I believe Vootele is directly familiar in using this system, and it seems like a nice system. It has a couple of cognitive memory testing, but very limited. So just to mention one that is most frequently used in the context of measuring memory is place learning and place relearning, and it can be both positively or negatively reinforced, and there are some already work using these IntelliCages for test in Alzheimer's disease mouse models. So they can work pretty all right, but for us, it was still very limited ensemble compared to the ones that I showed you before. And I guess I have to stress what I did not mention before is that each of these tests in this range of 8-week testing, it's important because they are not fully redundant. So for example, novel object recognition test is more sensitive towards rhinal areas. So perirhinal, lateral interinal areas, which is where Alzheimer's disease starts. And then something like a TMACE is more sensitive to hippocampus, possibly medial interinal areas. And then, you know, there are other aspects which change as well. So it's important to have more and sensitive and specific testing paradigms. And you can also notice that in this case, I'm not sure how necessary it is, but at least in this paper, which was reporting a study in AD mouse models, there was a quite significant water restriction. So the water was allowed only between 9 p.m. and midnight, which is essentially, 4 hours or so, which means that for 20 hours, the animal did not have access to water. And at least in UK, that would be considered a severe condition in your project license, and that's not desired as well. And that would, again, have some physiological consequences, which we wanted to avoid. So in that kind of context, we went on and developed our own system. And by we, I would like to acknowledge these 3 people who really were absolutely key people in the whole development, both hardware and software. So Hins Ho, I believe he is here now, Nate Kaysar and Marius Boja from UCL. And Hins and Nate are my PhD students. So this is what the Smart-Kage looks like. It's about, you can imagine it's, so the actual pictures of the first prototype is on the right-hand side. So you can imagine it's a transparent box of 50 centimeter, roughly speaking, 50 by 50 by 50, where the animal lives. Well, as long as we could test it. So I will show you in a sec, but our longest experiment was more than a year long continuously. So animal lives here. It's a image processing based system. So above this box, a cage, there is a camera, infrared camera. There is autonomous lighting sources. You can see them here, which means that the system really does not require any specific lighting condition. It does it for itself. And the animal is filmed in perpetuity for as long as experiment lasts at 2 hertz per second. And that is enough for our purposes for this cognitive phenotyping, this kind of frequency, which means that your data quantity is still manageable compared to in some applications where the frequency of recording has to be, well, above 60 hertz even. Okay, so there are 2 important mechanical or hardware parts to this system. One is these 2 drums, by which essentially novel object recognition and object in place recognition tests are implemented. And also these 2 corridors with the partition, which are also essential for implementing TMACE. And then in addition, we have some motor assays available to us, which, you know, are equally important. I mean, not equally, it depends on what you want to ask, but they are very important to have in parallel with cognitive testing, because we have to be able to rule out that some behaviors which appear like cognitive impairment are not really a motor impairments. So that's one thing. And also some animals do have motor phenotypes, specific motor phenotypes, which we would like to be aware of. So for that purpose, we have a running wheel, which we also characterize, nest. So it's important as well for characterizing quiescent behaviors. We had a ladder as well to see, to look at the climbing behaviors, which we did not really describe in the paper, still working on it. Okay, so that's the system. As I mentioned, it had the camera, it has its own microcontroller, which makes sure that the imaging of animals' behavior, and all the hardware is synchronized, and it has also its mini computer, each of the smart cages. And this is the working flow, essentially, that as I mentioned before, the animals is recorded at 2 Hertz per second. Then the data, this imaging data, is analyzed using a deep lab cut actually based algorithm. Then the essentially skeleton, a posture of the animal, is run through a bunch of classifiers to be able to say whether the animal was exploring, or the animal was still and sleeping, or the animal was under the ladder, or any other behaviors of interest that we would be interested in. And at the very end, I'll show you also that we use agglomerative clustering to phenotype the animals, to put individual animals into groups, so to speak. And I'll speak about it in a moment. And this is what the filming through this infrared camera looks like in 9 different cages at different times. So hopefully what you'll see is that they are indistinguishable. And this was really at the core of what we were trying to achieve, because what we wanted to do, we wanted to train our library to classify all these behavior and monitor the animals, but we would like ideally to do it once, so to speak, for all of the systems, and we don't have to go back and retrain and do something else. So this is all very, very standardized. Okay, so that's what we did. As I said, as I mentioned before, these classifications are based on a deep lab cut, and we verified how accurate we were using ground truthing. So the actual experimenter, human experimenter, would go through these hundreds and hundreds, in some cases thousands of images, annotate them, and we would compare the performance of our algorithm, completely unsupervised, to the human annotator. Well, in fact, 2 human annotators. And we wanted to make sure that each of the behaviors has 80% or more accuracy. That was our criteria. And of course, we reached that, and your accuracy really depends on the number of frames and the variability that you use for training. And this is just examples of the behaviors and how they look like. So this is the T-maze. I will explain how it works in a second, but it's essentially implemented by these corridors, as I mentioned before. Novel object, so you'll see how here, the animal, you saw the exploration of the panel. Running on the wheel is also done by images. Quiescence, the animals in the nest. And this was quite interesting behavior that we noticed only in the hippocampal animals, where they spin in these circles like a thigmotaxis. It's also called wall hugging behaviors, but you can see that in the home cage environments, it's not so much a wall hugging as a stereotypical running in circles, not necessarily around the walls, but around probably some obstacles, some things, and etc. But still very stereotypical. Okay, so let's move on to the actual task. So I'm sure that many of you in this audience knows how the standard forced choice alternation T-maze task works, but I still will explain it briefly so that we're on the same page. And I would like to say also that this is probably one of the most sensitive hippocampal tests that there is. And it turns out from our tests and actually from other tests as well that I could find in the literature, that it's not so many of the reports, but still the ones that I was able to find is also medial entorhinal or entorhinal cortex dependent task. So the way it works like this, the animal is taken out of its home cage, brought to some kind of experimental lab where there is a T-maze. So there is 2 corridors stuck perpendicularly. And in the so-called sample phase, the animal, one of the arms is blocked, and the animal goes to unblocked arm to receive the reward. And then immediately after the animal receives the reward, it's brought back to the beginning of the stem of this T-maze, but now there is a door there, so the animal has to stay in this initial segment of the T maze for X amount of time, which is called intertrial interval. So it's usually, it can be anything from immediate, like a few seconds, to minute to a few minutes. And the longer it is, the more difficult it is for the animal to solve it, to solve the maze, to choose a correct choice, and the more hippocampal dependent that test is. And so in the test phase, after this intertrial interval, the door is released, opened, and the animal now can run to both left and right. And to get the reward, the animal has to choose the corridor, which is the different one from the one he or she just visited, right? So if it went to the left-hand side, now it goes to the right-hand side. So in order to know how to do this task correctly, the animal has to remember its previous choice. And that is very much hippocampal dependent. So in a Smart-Kage, what's so-called Smart-T-Maze alternation task, not to confuse it with the standard one, what we did is basically, it's a very similar concept. The animal has to alternate between the arms in this Smart-Kage to receive water. So the water is not restricted. The animal is not, there was absolutely no deprivation of water control, as I just pointed out in this previous study where it was 20 hours unavailability or something like that. No, the water was fully available. It just, the animal has to, in order to get it immediately, the animal would have to remember its previous choice. And that was the sort of a profound revelation that we were able to observe, that on average, so given enough N's, of course it's noisy, but given enough N's, this negative reinforcer of not finding or getting the water immediately was good enough to make sure that the animal learns this task. Okay, so the animal has to basically alternate. And so you will notice two things or two differences. One is that unlike the standard T maze, in which animals are always usually food deprived, not water deprived, at around 85% restriction of its original weight. In a Smart T maze, we didn't have to do any of that. There is no need for water or a food restriction. So that's number one. Number two is you can immediately sense that the animal engages with the trial when it wants to drink. And that means that we have a whole sample of different ITIs that we can measure. And we will get back in a second to that when I show you the results. So that's how the Smart T maze works. Okay, now novel object recognition. Again, just a very brief reminder what this task is in a standard protocol. So in the standard protocol, the animal is taken out from its home cage environment into laboratory room and is then placed to some kind of relatively small, usually square enclosure, and is presented with two objects that it has never seen before. And the range of these objects really varies between different people, different laboratories. So the animals tend to explore these objects. Okay, so then after X amount of time, so usually a few minutes, and usually there is some kind of a cutoff time that they must explore this object minimally for the task to be included, they're taken out of this enclosure for X amount of time. And that's, again, some kind of intertrial interval, which can be a few seconds to a few days, usually. And then the animal is brought back to the same arena with one object being identical to the familiar one, and then and so normal animals normally tend to explore the novel object more. And that's a signature of a memory of the old object. And that test, it is non-spatial in this arrangement, and it's very much dependent on the perirhinal cortex and lateral entorhinal cortex. Sometimes some groups report that it may be dependent on hippocampus as well, and we think it depends really on the strategy animal is using to solve it. So in one case, the animal can use familiarity. So like, I've seen it somewhere. I don't know where, but I've seen it. And that would be familiarity strategy, and that is probably rhinal dependent, at least from what we know in humans. Or the animal may use a recall strategy, which is probably hippocampal dependent, where the animal is actually sort of remembering, so to speak, an event of encounter with these objects before, and then uses that to identify the new object. Object in place is very similar, but instead of changing the familiar object, one of the familiar object to a new one, the location of the familiar objects is changed, and that is hippocampal dependent. And as I mentioned before, the longer ITI is, the more difficult it is for the animal to solve it. And again, it could be that will determine which strategy the animal would end up using eventually. And this is a very much like test. It's probably the most often used standard test in memory research. It's very simple. It's naturalistic. It uses this naturalistic tendency of the animal to explore novel objects and so on. So it's a very attractive test to use. However, I have to say, in our experience, and also from the feedback that I got from many labs which do use cognitive testing, it's actually very, very difficult tests to implement in a standard way, because it depends on all sorts of parameters which people tend to vary. It depends on how much animal is familiar with the actual place where this experiment is happening. It also may depend on the objects, or it actually does depend very much so on the object that I used in this experiment, and that varies a lot. So in our hands, we found it very difficult test to implement. And for that reason, again, we wanted to really automatize it as well. So the way it works is the objects essentially become patterns, which are presented in the Smart-Kages via these windows that you saw, you may remember before from the picture that I showed you. And through this window, these patterns, they can be touched, smelled, interacted with. So everything that the animal would normally do with the object, unlike presenting them, for example, just visually on the screen, where if there is a visual deficit, that could be mistaken for memory impairment, something like that. So this is exactly like what you would get in the standard case. And so we have a cassette, a bunch of different patterns that we can present to the animal at any given time when we want. So we can choose when we start the test, how long the sample phase will last, what is the ITI, what is the test phase, and all of that completely unsupervised. So you can run it, we could run it at night if you wanted to, without the experimentalist being present. The important bit is this, that of course, when there is the change happens, so rotation of this drum happens to change from one pattern to another pattern, the animal is inside the corridor so that it doesn't actually see and respond to the rotation of the drum, rather than noticing the novel pattern as novel, for example. Okay, so hopefully this is clear, and if not, please do interrupt me. But these are essentially those cognitive tests which we really wanted for our lab to study hippocampus and entorhinal cortex. And this is the protocol that we use for the pilots to see whether those tests actually do what they are supposed to do, from what we know from standard testing. And we tested different groups of animals. So animals with hippocampal lesions versus what they did before they were lesioned, medial entorhinal cortex lesions versus pre-lesion periods of those same animals, sham lesions, which means that we injected in the procedure of the lesion is exactly the same, but instead of using ibotenic acid, we used saline. And we also, as a proof of principle, again, all of those are very small samples of animals to start just as a proof of principle for this method. We also used amyloid beta knock-in animal, which is probably one of the most used for AD research. And we used it in 2 ages. And what we would normally do is we would measure or phenotype these animals in the Smart-Kage for a month or so. Then we would do the lesion. And then we would phenotype again for 8 weeks post-lesion. And then we would take these same animals out of the Smart-Kages and do the standard testing outside the home cage environment so that we can compare the performance on both of them. When it comes to APP knock-ins, of course, we didn't do the lesions on these animals, but we tested them at 2 different ages. One was around from 6 to 9 months. So there were different ages of these animals, these few animals. And then after almost half a year, we retested them again in the Smart-Kage. Okay, so this is just to show one animal. It's an example of one animal which has been tested for 12 months continuously. This is its performance on a T-maze task. And we spread it for different ITIs, as you can see here. And I'll explain in a second why those different ITIs may be of interest. This is already not a single animal, but a group of 5 hippocampal lesion animals. So this is pre-lesion performance, and this is engagement with the spout. And this is post-lesion. And this empty white period segment just indicates that they were recovering outside the home cage environment just after the lesion for 7 days, and then they were put back. And as you can see that this was like really strong effect on these T-maze task. The performance of the smart T-maze just dropped like a stone in hippocampal lesion animals. Okay, so this is where, and you can see that the compensation starts at some point, but many months, well, a couple of months later, it kind of starts ramping. And this is actually quite similar to what you see in the Morris water maze as well. So there is some kind of patient who starts later on, and they probably start using some kind of other clever strategies or something like that. And now you can see the performance of these animals pre-lesion, which is in the blue, and post-lesion in gold, for different ITIs. So remember, I mentioned to you that the animals can choose when they want to engage with the task, and that is in a way good for us, because we can then see what was the performance at different ITIs. And if we consider 50% performance being a random chance, you can see that they perform at randomly about ITI equal to 10 minutes. And this is actually what people reported, researchers reported in standard tests as well. So it's always up to 10 minutes. And it also drops exponentially in the standard testing. And it was not the case for control mice. So this is pre-lesion and post-sham. You can see that they are indistinguishable. And just to make this may be interesting for like a future slide, the important parameters that we found in this curve, amongst others, things that we could quantify, is the maximal performance that they can reach, so 80% pre-lesion, and also the time it takes to drop to the randomness, which we consider like a memory span. So those two things may be changing depending on the phenotype. One that was particularly interesting from our lab's point of view, given our interest in medial entorhinal cortex, that there was quite a big difference between performance of ME size. So you can see here on the right-hand side, hopefully you can see my pointer, that in a standard case, these are the same mice, right? So in the standard T-maze, these mice were quite impaired. They were almost as bad as the hippocampal mice, with the exception maybe of one, which is slightly bigger, still worse than controls, usually. Whereas in Smart-Kage, they were quite on smart T-maze, and that took us as a surprise, and we're still investigating. One thing that I should mention, you can see this point here versus 3 points here, and I would like to stress it's a very small number of animals, just as a proof of principle. But when we install it for these mice, this, the only animal which had a full MEC lesion, all of these were partial lesions. So for the partial lesion, these animals were not in a smart T-maze versus it was still big on a normal standard T-maze. And as of yet, we don't know why. Whether that opportunity for us now, two of these tests side by side, we can actually distinguish almost with 100% accuracy each of these animals' identity. So it's not only like a group average effect where people are speaking, okay, on average, controls performing better than HPs. No, we can take the results of this test and we can go animal by animal and correctly to the group. And that is actually quite important for the basic research studies that in our lab we would like to be able to identify animals at different stages of their diseases. So if we can pick animal with a high accuracy on a roadmap of the disease and do some like treatment on it or some testing which is, say, more expensive, like MRI scan or 2-photon imaging, that would be very beneficial for us. Okay, and this is just quickly to show you what we saw for APP and LGF mice, so amyloid beta Alzheimer's disease models. And similar to MEC mice on maze, the maximum performance was not that much altered as they went older, but what seemed to be getting worse is the memory so-called span. So how much they could maintain the relatively high above chance performance. You know, what is long TI that they could maintain. And that was different. It was not significantly different, but it's mostly because of the very small sample size, we think. Oh, no, object recognition. This is what we did for this initial article that is published, so you can look at it if you wanted to. Just a short recap. Essentially, what we did is we changed the panels, as I described you previously, and we wanted to see if that would trigger increase in exploration of the changed panel within the animal. And the short answer is yes, it does. So the exploration of a novel object increases. I'm not showing it here, but it is in the paper and you can have a look at it. And if we look now into what is the exploratory patterns in these different animal groups, you can see that, well, it's non-significant from any of the groups. That again, I think is because of the small sample size. It seems like it's increased in the hippocampal animals. We also notice this very periodic thing, which may be an artifact of a small group, but we are very keen to investigate what is this long-term periodicity patterns. And then when it comes to the APP and LGF, it was actually quite a robust effect to decrease in novel object exploration with age. But as you could sense, there was no ITI, there was no memory test per se, or at least not in the way it's usually done in a standard way that I described previously. So now we are preparing an additional study just on the novel object. I'm very keen to share some of the unpublished results with you as well. So in this case, now we have a test phase during which we change what we now call fixtures, so some kind of patterns on the wall paintings you can animal lives with. So on the actual novel object recognition task, we can change new novel patterns. Of course, when the animal is outside the open arena, Smart-Kage. So that lasts for some X amount of time, in case 24 hours, which is the sampling phase, analogous sampling phase. And then it changes back to the so-called fixtures, the paintings, familiar paintings on the wall. And then after intertrial case equal to 24 hours, we present the animal with the identical pattern that it's seen before, not the same. I would like to stress to avoid confounds of many arcs that the animal may have left. We also present animal with a complete new pattern, and we exploration looks like. And there are well, two important parameters that we wanted to achieve for this task to work. Each of these patterns, if they both of them or all of them are novel, should be the same. That is like the baseline of any good standard novel object recognition task, and that's the first thing that we had to verify and search for these kind of patterns, which was not easy. But we found them, so in metal, wood, and type of acrylic, and you can see that the exploration time is non-significant between them. It's also balanced between the left side and the right-hand side. And you can also see that reflected in the discrimination index. So you can see that during familiarization phase, animals are equally likely to explore both of the objects, and all of these patterns were balanced between the sites and whether they are novel, whether they are identical, etc. So all of that was automatically balanced. And then when we during the test phase, when we compared the exploration of the novel pattern compared to the new, familiar pattern, you can see a really nice, strong, significant effect. So the animals really tended to explore a novel more than they did the familiar one, as described by discrimination index. So this is very exciting. It's just one we are preparing the full manuscript on that. Okay, and just for the models, which are much more commonly available in all sorts of other systems and use of other messages, but one of them is way sense. So how much animal is non-moving, so it's sleep per se, but there is another paper by somebody else which show that it's actually a very good proxy for sleep, at least in a black 6 animal. Obviously, if your phenotype is some kind of moderate, may not be the case, be really established before making this sort of conclusions about versus quiescence. So I'm just calling it quiescence. And this is an example program of one animal, pre-lesion and bottom post-lesion. That is a particularly beautiful animal. So the white parts is segments non-moving in this case, so light cycle, the animals, right? And the black show the times during the day when the animal was moving. And you can see that this animal, this specific animal, really had these very nice regular naps, especially towards the end of its active day, to speak. And it was waking up, and we looked at the videos. It's very nice. It's usually for going and drinking at that point. And then after the lesion, you can see that this hippocampal animal tended to wake up much more frequently, and it also it's nocturnal naps, so the active phase naps were also kind of disturbed compared to the regularity it enjoyed before lesions. And this sort of change in mean daily quiescence during light phase was just, it was a strong effect across all hippocampal animals, and there was no significant in controls and MEC animals. And the sleep changes shown here on the right-hand side for APP was actually one of the most prominent changes that we saw in these animals in the whole feature domain. And you can see that they are changed here as well. We also quantify locomotion parameters. So it is known that from the standard test that hippocampal animals tend to be hyperactive active. This is what we found smart. Quite interestingly, it did not translate into the wheel, even though they were hyper intended on the wheel, and no change in and no change in MEC animals as well.
Sorry,
Julia.
Sorry.
And for APP and LGS, as we well tended to move less. That's what we found compared to themselves when they were younger. Yeah.
Sorry, Julia.
I'm sorry for that, for the connection problem. So basically, pre-lesion, this specific animal, this is one example on the left-hand side. You can see that it had this very regular napping during the active phase, towards the end of the active phase, which collapsed after lesion to the hippocampus. And also what you can hopefully see that during the inactive phase, so the light phase, the animals started waking up much more frequently. And that was the case. This is one example, but that was the case for all of the animals that you can see in this middle histogram bar. You see high significance in the mean daily quiescent time in the hippocampals, but not in sham controls and not in MEC animals. And we could also see that the changes in quiescence patterns in APP and LGFs, and those were one of the most prominent features as the animals were going older, which was not observed in the controlled APP mice. So they're just wild types, which means that it's not just an age thing to become slower and older and, you know, change in quiescent states. Now, for the locomotion, so I just mentioned that it is known from the standard test that hippocampal animals tend to be hyperactive, and this is exactly what we measured as an increase in mean daily locomotion in hippocampal animals in the Smart-Kages as well, but not in other groups. And what I was saying is that interestingly, this pattern did not translate into running on the wheel pattern, which decreased in hippocampal animals, even though they were hyperactive. And you can see one example of the ethogram on the left-hand side. And there were changes in locomotion, APP, and LGF as they aged. They moved less, whereas it was not the case for the control age-matched control animals. So it's not an age thing. And this is just the same figure, but sort of expanded or unpacked across days, just to show you how stable these changes are across days when we measure it for 30 days pre-lesion and post-lesion in all of these animal groups. Okay, and then the final slides, what I will show you is that, well, just going back to the problem that we were trying to solve, because it's not like we wanted to invent a new home cage monitoring system. We had a very specific purpose from the point of view of the lab, what we wanted to achieve. One, we wanted to scale cognitive phenotyping, and the only way to go is not to have a human experimentalist. So that was very clear to us, and we achieved that. The second one was that we wanted to have a sensitivity which would allow us to stratify animals on individual basis. So in Alzheimer's disease, we are interested in studying circuitry, and therefore we would like to start measuring that circuitry just on time. And the way it is done right now, it's like on average. So we know from the histology where pathology should be, like, you know, 6 months nothing, or some hypophosphorylated tau, 12 months some accumulation of some proteins of interest, for example, and etc. So those ages would be chosen on average, but anybody who works with these different groups of animals know that there could be some very significant variations in the pathology, even in the same sort of cohort, the animals which are siblings, and for any practical purposes should be very similar. They live together, the same age, the same gender, but they may be quite different. So we really wanted to identify where in the disease they are based on their behavioral phenotype on an individual animal level, not just a group effect, and then perhaps choose those animals at these given time points for further investigation, like looking at circuitry properties, imaging, MRI imaging, anything that otherwise would, you just can't do it for the entire cohort. It's not practically possible. And for that, we took all of these behaviors that I just talked about, both cognitive and locomotion type of, so passive and active. And we came up with quantifiable measures for each of these behaviors. For example, so this is all of them that we use, 32 dimensions, but just to give you an example, take a performance on a T-maze, we would take this highest performance that I already mentioned and the memory span, so i.e. when the performance dropped to 50%. And et cetera, et cetera. So for all of these features, we'll make a multi-feature space, and we would then cluster each animal in that space. We would assign the animal to specific clusters. And again, it's all described in the paper. So for the sake of time, I'm not going to go into details of that, but I can just tell you that what we found that using this multidimensional feature space was much more effective way of pointing out to a correct phenotype on individual animal basis. And this is what you can see here. This is a confusion matrix on the left-hand side, and you can see that, yeah, hippocampus, it's such a strong effect, we can do it 100% accurate. We could say these animals hippocampus, this, and we see much more difficult. APP was quite good, actually. It's a very small number of animals, but this was comparable to what we could achieve using the standard testing, for example, without actually doing any tests, as you could imagine. So we really think then, as a result, these kind of tools is a way forward. So just retrospectively, I think it's important to acknowledge it's not only the image recognition that is allowed, that is required, sorry, to allow these kind of phenotyping, unsupervised phenotyping, it's also hardware, and those two have to be just absolutely perfectly working together. We, as a proof of principle, we showed that these kind of systems can be used effectively for cognitive and locomotion related behavior classifications and characterizations. And also, using multidimensional feature space, we were able to accurately, so with high accuracy, stratify animals, so identify individual animals, assign them to the group, which would be for us very useful for our further experimental pipeline, and was really a requirement that we wanted to achieve. So on that note, I would like to finish this presentation. I would like to thank everybody in the lab, but especially Hinz and Nates, who really led this study, and Marius Bourgeat, who is actually working at UCL with John O'Keefe, who was absolutely instrumental for this study implementation, and then Bart de Strooper, who was our Alzheimer's expert in the whole study. And of course, funders, Wellcome Trust, UK Dementia Research Institute, and others. Thank you very much.